Right, good morning. Thank you for being on time. We have a lot of things to do today. We always do, but it's the second last week, so hang in there. It's almost done. Number nine is more difficult than number eight. I am so sorry, okay? Uh, I will try my best. The difficult stuff, um, I don't know, maybe the more difficult portion is the second half, right? And it's gonna happen after the quiz on Monday, so I'm a little bit worried. But
Wait, this
see
is
what.
gonna be on the final, but we're not gonna have a test quiz.
Yeah, this is gonna be on the final. Okay, this is not on your quiz on Monday, obviously, but this will be on the final. And then number 10 is easy. Number 10 is a walk in the park.
If we,
And I won't be able to finish all of number 10, so it would just be like maybe one third of it. And it's gonna be fun. Okay, but before we do the fun stuff, we have to do number 9, which is biotechnology. What is biotechnology? Biotechnology, but is basically technology based on biology. It sounds very simple. Essentially, after learning all about all the cellular processes, scientists try to control these processes. They try to alter it and ultimately harness the power of these cellular processes to create new technologies that will benefit human lives. All right. In the second half of the course, in the second half of the course, we learn about DNA replication. We learn about transcription, translation. So how do we use our understanding of these things to create new technology that would help improve healthcare, improve agriculture, or improve you know other forms of our lives. That's basically the goal of biotechnology. There are obviously many different examples of biotechnology. Some are very difficult to understand, and we don't have time to go into all the details in just 2 lectures. So I selected a few to show you guys. The first one we will be talking about extensively, probably going to take us an hour, is recombinant DNA. All right. As always, I'm not going to ask you, well, not as always, but then like in this particular case, I'm not going to ask you to explain the whole process, but you have to understand these processes in order to answer the multiple choice question as well as the application questions. Okay. The application questions, like I said, are difficult. So please pay attention. What is recombinant DNA? Recombinant DNA basically is a chunk of DNA that is stitched together from multiple sources. Right? So you can have DNA from mouse, and then you can have DNA from jellyfish. You put them together. Now this is called a strand of recombinant DNA. It doesn't have to be 50-50, okay? Even if you just insert a little bit of a jellyfish gene into, say, the mouse, it's already recombinant, okay? You can take human DNA and put it into a bacteria. Now you have recombinant DNA. All right. The mouse picture right there is not a Photoshop picture. They actually can create mouse that glow in the dark. Okay. They are called GFP mouse. Where's my pen? What they did was they took the glowing gene from jellyfish. It's called the green fluorescent protein. And then they put it into the mouse genome when they are still like in the embryonic stage. And then the mouse, every cell in the mouse glows in the dark. They don't do that just for fun. There are useful application of that, but we're not going to go into the details. If you're interested, you can look up GFP mouse. So recombinant DNA are useful in many settings. I'm going to give you 2 examples. I will start with its agricultural application, and then we're going to spend the majority of the hour talking about an application in medicine. Everybody understand what recombinant DNA is? You just take DNA from different organisms, put them together, then you have recombinant DNA. All right. Wiki, guys, stay with me. Golden rice. Anybody heard of golden rice before?
Oh,
Most people have not. That's okay.
and then,
So, yeah. Golden rice is an example of a GMO. GMO stands for genetically modified organism. I'm not here to tell you whether GMOs are good or bad. I'm just telling you how it's being used, and you can, form your own opinions. All right. So vitamin A deficiency is very common in low-income countries. Right? It is, in fact, a leading cause of preventable childhood blindness. It turns out the vitamin A is needed to create a particular molecule for the eyes in order for you to see. So if you don't have enough vitamin A, that could lead to blindness. Vitamin A deficiency also lowers your immunity. And in countries, low-income countries, they often do not have high vaccination rate either. So lower immunity plus few vaccination together can cause an endemic of infectious disease to happen. We are all familiar with pandemic is the whole world, okay? Endemic is like a localized type of a problem, okay? And then one level higher would be like an epidemic. Okay, so something like malaria would be like regional, that's epidemic. Endemic is like really, really localized, okay? In some countries, each year it is estimated that vitamin A deficiency is related to about 670,000 deaths in children under the age of 5 and cause an additional 500,000 cases of irreversible childhood blindness. So obviously this is not nothing and it's quite tragic. And if we can prevent childhood in a blindness in a child, why wouldn't you do that, right? This is a heat map that shows you where the vitamin A deficiency is a severe problem. As you can see, not so much of a problem in high-income countries like Canada and the US, and other places, but it's quite a problem for low-income countries. So that's where golden rice comes in. Okay, golden rice is a genetically engineered rice that is created to make something called beta carotene. Beta carotene is a building block for vitamin A. If you get beta carotene into your system, it will be converted into vitamin A, which then can supplement the vitamin A deficiency. You probably have heard this before. Either your parents tell you this or you tell your kids, eat more something because it's good for your eyes. What's that something?
yeah.
Exactly, right? Is that true? Well, maybe there is a little bit of truth to it because carrot, being orange, has high levels of beta carotene, right? Which then actually allows your body to convert it into vitamin A, and then it's gonna be good for your eyes. Okay, but generally speaking, if you are living in a wealthy country where food source is generally not a problem, you wouldn't really need that extra bit of carrot. That little bit of extra carrot is not going to give you like super good vision, right? But it might make a difference if vitamin A deficiency is a problem. But here's the thing: you can't just ship a bunch of carrots to countries that are suffering from vitamin A deficiency because number one, it might not have a very long shelf life. Okay, it can turn bad really quickly. And how much exactly do you need to eat in order to prevent the problem, right? Access is also quite a bit of a challenge there, right? Not everybody can afford some of these food that would supplement their vitamin A. And so, making a genetically modified rice that has beta carotene in it seems to be a logical choice. Not only is rice part of the staple food of a lot of these low-income countries having the problem, they are also quite easy to ship and store for relatively longer periods of time. Okay? So what does this have to do with recombinant DNA? Well, regular rice do not have beta carotene, okay? The white rice, the brown rice, they don't have any of the beta carotene, and you can see the golden rice here actually does have it, and the color is different. The way to do it is they actually took genes from daffodils, which are, these kind of flowers. There's a gene called Psi in it. I never mind what it does. And then when we write genes, we usually write it in italics to show that it's gene. And then with another gene called CRTL from soil bacteria. And when you put those two genes into the DNA of rice, they start making beta carotene. Okay. People who are proponents, they support the use of golden rice. Here are their point of views. It addresses the problem of vitamin A deficiency. It will improve public health because don't forget, the vitamin A deficiency is linked to lower immunity. So if you're able to supplement that, you will improve public health and hence food security. But not everybody is on board with this idea. Right? Opponents of golden rice argue that there could be unintended consequences. Right? They argue that we don't know the long-term effects of the product of something like GMO on the human bodies. It's not just the body that of a person that they're worried about. They're also worried about the environmental impacts. What if these things are grown and then it got transmitted into the wild? Is it going to affect the growth of other crops? Is it going to become an invasive species, for example? They are also worried about corporate control and patents, right? They worry these biotech companies would have too much influence, okay, in the places where these golden rice are being distributed, and also things like socioeconomical factors, access, inequity, so on and so forth. So both sides has their arguments, and you can look more into it if you're interested. That is recombinant DNA applied in the field of agriculture. And by the way, this is not experimental. Okay, it's already out there. Right? It's being used to target people who have vitamin A deficiency in countries where access to vitamin A is limited. Okay, you guys understand? Do we have golden rice in Canada? No, because we don't have the problem, right? Wonderful. Number 2, we're going to learn about the application of recombinant DNA in the context of medicine. Insulin. Insulin is a hormone produced by the pancreas. Do you know where your pancreas is? Can you show me where your pancreas is? Just point to it. Which side is it on? On the left side, right? Okay, it's on the left side behind your stomach, okay? About this big, okay, and then it produces insulin. Okay? So insulin is important for blood sugar regulation. When you eat a meal, you get carbohydrates, you get sugar, you get glucose into your bloodstream, but if it stays in the bloodstream, you cannot use it. It needs to get into the cell. And that's what insulin is for. Insulin will encourage the glucose from the blood to go into your cells, and then you can use it for cellular respiration. There are different kinds of diabetes. Okay, the one that you are probably thinking of is type 2 diabetes. That's related to unhealthy lifestyles, obesity that tend to occur later on in life. That has nothing to do with what we're talking about. Right? People with type 2 diabetes, you cannot give them insulin. It's not going to help them. Right? Type 1 diabetes is something that usually occur earlier on in life. If not, the person might be born with it. And people with type 1 diabetes do not produce insulin. And they need to inject insulin in order to survive. Okay? So before we are able to use biotech to mass produce insulin, the only way to get insulin is to extract them from cows and from pigs. Okay, the scientists who discovered insulin are actually Canadian scientists, Benting and Best. They have like a building to honor their name in downtown U of T, right? And the only ways to get it was to extract the pancreas from the cows and the pigs, grind them up, and then get the insulin and give it to people. What might be some of the problems with that? What are some disadvantage of getting the animals insulin and giving it to people? Anybody?
They could have disease.
Yeah, so like you might worry about the purity of it, right? When you extract it, like it's very hard to ensure there are no impurities. What other problems? Like what if I give you insulin tickets from cows and pigs? What kind of questions are you gonna ask? No questions, just jab it into me.
Where are the pigs from?
Okay, but what about that? Well, if it's from, well, I guess you have, if it's from pigs, right? Because of religious reason, it might not be suitable for everybody, right? Okay. Another problem is the cost as well as the process to extract it might be expensive and lengthy. And also the animal version of the insulin it is not the same as the human version, right? So the effectiveness might not be 100%. Right? So in other words, it's not ideal, right? But that was the only way to get it. You can't get it from other humans, right? Like that's just unethical and not possible. That's all they had until the biotechnology was available, and in the late 20th centuries, we were able to hijack the bacteria and force them to, force is a strong word, I guess, and make them turn into insulin factories, basically. Okay. So what we're going to do in the next hour or so is to learn how to turn a bacteria into insulin factories. Okay? Here we go. There are 4 steps. Number 1, we have to locate the gene of interest. There's so many genes in the human genome. Where is the insulin gene? Right? We have to find it first. After you find the gene, you have to clone it. You have to make many copies of the insulin gene. You don't need all the genes. You just need to copy the insulin genes millions and millions of times. Then you will take all these insulin gene and you feed it into a vector. A vector is something that will take the gene and make the product. In this case, bacteria. And then finally, once the bacteria accepted the gene, right, then it's a recombinant DNA because it has bacterial and human DNA in it. And then you let nature do the rest. So how do you know if bacteria has taken up your DNA? Well, one way to do it is to give them a special dye, okay? I'm not gonna go into too much detail, but basically this dish here is about this big. It's a petri dish. You grow bacteria on them. And these are the white ones and the blue ones. They are bacteria colonies. Right? If you can see it with your naked eye, that means there are millions of bacteria in this particular spot. Okay? You literally can take a hoe and scoop them up, and then you dump it into a new plate, and then it will start growing. Right? So the blue ones are the ones that has accepted the insulin genes. Those are the insulin factory producing bacteria. Right? So we'll scoop the blue one out and then, mass produce them. Right? So and then we leave the white ones alone. What?
The vector separate.
That's right, they accepted it. Right? So this is a visual way to tell, right? But that's not the only way to tell whether it has accepted the DNA or not. We're going to talk more about it later on. Okay? So 4 steps: locate a gene, clone the thing, insert it into the vector, the bacteria, and then nature will do the rest. Let's start with locating the gene. That used to be a very lengthy process. Okay, you to search for the gene might take you decades of research, right? But thanks to the Human Genome Project, which started in October 1990, and it took over 10 years to complete. It was an international collaborative effort to map out all the ACTGs in the human genome. Okay, they want to know what are the nucleotides that makes up each of the chromosomes. That's the goal of the project, right? And once it's finished, eventually they publish this online, and nowadays we can just go on the internet and find any gene you want. Okay, so I'm going to show you how it is done. First, you need internet, and then you're going to go to this thing called, you just Google Human Genome Browser, okay? And you are going to key in the gene that you want. And I did BRCA1 gene because that's something that we learned, right? You guys remember BRCA1 gene? Yeah, breast cancer gene 1, right? Okay, it's a tumor suppressor gene, right? So I'm going to search that and we're going to go. And there you go. Where is the BRCA1 gene? BRCA1 gene is on chromosome number 17. Okay. And over here, that's the centromere. The stuff before the centromere is called the P arm. The stuff afterwards is called the Q arm. So it's located right here. Okay. And in terms of base pair, it is base pair. I don't even know how to read this number. 43,044,295 all the way to 43,125,364. That's how long the gene is. Okay? If you click into the gene, you can find its sequence. All right, so I'm gonna submit, and that's the gene. Okay? They did that for all the genes in the human bodies. Okay? And since then, they actually do it for a bunch of other organisms. If you want to look up genes for your dogs or cats or chickens or whatnot, it's available on the same website. Okay, so now it's very easy. If you want to clone a gene, all you gotta do is you're gonna find where it is, and then you move on to the next step. Do you understand? Okay, this is real stuff. You can actually convert this into RNA and then convert it into proteins because I taught you how to do the transcription and translation, right? Okay, so the stuff we learn is not fake. Okay, it's real. There are real implications in medicine and in agriculture and whatnot. Okay. All right. Gene has been located. Next step. What are we gonna do? We're gonna clone it. We're gonna copy it millions of times. All right. And we want to do it fast and do it quick. Before PCR was invented, cloning a gene takes quite some time. Right? And then PCR was invented. You have heard of PCR, right? During the pandemic, maybe for the first time? PCR testing, anybody hands up? No, really? Okay. Like you do the swab, they send the sample to do a PCR test for you, right? And that's what happened at the beginning of the pandemic. I mean, they still kind of do it now. But then later on, they came up with the kit for you to take home and test it, right? Right? The one that you can wait for the 2 bars or 1 bar to show up, right? Right? So the take-home kit, that's for protein testing, right? Testing for virus protein. Yeah.
Wasn't it for like, to see if you're immune to COVID or not?
No. The PCR test for the virus genetic material, okay? As opposed to the one you do at home, that tests for the presence of the virus protein, okay? My point is this though, PCR was actually invented way before the pandemic, okay? And the purpose of PCR is to clone DNA. Okay, it was just being used as a way to test for COVID, which maybe we'll have time to talk about later. Okay, but PCR stands for polymerase chain reaction. If I ask you what is PCR used for, remember, cloning DNA. Cloning means make many copies of it. Okay, many copies of it. This is a PCR machine. Okay, if you walk in any research lab, they probably have a few of these things sitting around. You would prepare your samples in a little tray. It's kind of like those spot plates you guys use in chemistry class. And then you put it in a machine, you close the lid, and then you program it. And within 2 hours, you would have billions of copies of the gene you're trying to clone. All right. So it runs in cycles, which I'll explain in a second. But every time it completes a cycle, it doubles the amount of DNA that you have in the sample. So if you have one copy, and then after one cycle, you would have two copies. After two cycles, you would have four copies, 8 copies. It seems very slow at the beginning, but if you do the math, let's say you do it for 30 cycles. How much is 30 cycles, guys? I don't even have my, do I have my calculator here? Just plug it in, right? I got a calculator here somewhere. 2 to the exponent 30. Right? Over a billion, right? Okay. So in 2 hours, you're going to have over a billion copies of the gene that you want. So how does PCR work? PCR is based on our understanding of DNA replication. In DNA replication process, the first thing you have to do is to unzip the DNA using helicase, right, and gyrase, right? But instead of doing that, we will be using heat to separate the DNA. So you're going to heat up your sample and cause the two strands to separate, right? Around 95 degrees plus or minus, right? Depending on the CG content of your sample. If the gene you're trying to clone has a lot of C's and G's, you need the temperature to be higher because C and G's are more difficult to separate, right? Okay. If it doesn't have as much C and G's, then temperature could be a little bit lower. Okay. Once you separated the strand, then each strand, of course, serve as a template to build the other half. Okay. What enzyme is responsible for building the complementary strand? Thank you. DNA polymerase, right? Okay. But DNA polymerase cannot start the process on its own. Remember, it needs to have a primer, right? So that's what we need to do in PCR as well. You have to create primers and tell the polymerase where to copy. Remember, the entire genome is going to be in your sample. If you supply like cells into your sample, it's going to have all the DNA. But you don't want to copy all the DNA. You only want to copy a particular gene, right? Since you can look up the gene online, you can design a primer that would bind to the beginning of the gene as well as the end of the gene, So these primers would essentially flank the gene that you want to copy. And when the polymerase come, it will only copy things that are between the primers and not anything else. Do you understand that? It's like this. If I have a textbook, I don't have one. If I have a textbook and then I ask you to photocopy some pages for me, I put a sticky tag on one page and then I put another sticky tag some other page later, and you only copy everything between those tags. Okay? And not everything else. Because we don't want to clone everything, right? We only want to clone the gene that you want to make the product of. If you're making insulin, you only want to clone the insulin gene. Do you guys understand? Right? And the thing is, you can control what the primers are because you know the sequence of the gene. And so you just create, if I want to create a primer for this, then it will be like TGGAAG. It's complementary to this. Okay? And then same thing for the beginning. So in this picture, you can see where's my mouse? Oh, there it is. Okay? So that's the primer, and everything in between will be cloned. Next, we cannot use regular DNA polymerase 3. Okay? Regular DNA polymerase 3 is going to break down and denature when we heat it up to 95 degrees. Why do we heat it up to 95 degrees at the beginning again? To separate, right? Okay, so if we separate the strand and we apply a lot of heat, the DNA polymerase III is just going to break down and not going to work. All right. So instead, we use something called tag polymerase. You guys have seen these hot spring before? Okay. It's very, very hot, up to hundreds of degrees sometimes. But there are bacteria living in these hot springs, right? So what scientists discovered was that those bacteria has a heat-resistant polymerase, which makes sense because those bacteria needs to copy their DNA as well, right? And if the environment they're in is very hot, then their polymerase is not going to denature at high temperature. All right. So that polymerase is called tag polymerase. Okay. And that's the one we use in PCR. Okay. So basically we have the template strand. This is the section we try to clone. And we design the primers to flag the beginning and the end, and then the tag polymerase will copy. The PCR process is separated into 3 steps. Right? First step is denaturation, which is around 94 to 96 degrees. We use heat to separate the DNA. Once you have separated the DNA, you want to cool down a little bit. Not all the way back down to room temperature, because then everything would just close and come back together. You just want to cool it down a little bit, and that would allows primers to bind to DNA. And this process is called annealing. Because the primers, the pink stuff here, are complementary to the parent strand, they would find out where the locations are and attach to them. And finally, you would heat it back up a little bit. Okay. Remember, the polymerase is accustomed to high temperature. So at 68 degrees is not going to work. You need to heat it up a little bit to activate it. So you bring it to about 72 degrees. Like all these numbers are just like for your reference. You don't have to memorize it, okay? Like actually when you do the experiment, you have to use math to figure out, what's the optimal temperature. Like, obviously, we're just doing it very briefly. So this will activate the tag polymerase. Okay. So all this stuff would be in a tiny reaction tube. That's no bigger than what I'm holding here. Right, so it's really, 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 really tiny. You add all the reagents in there along with your DNA sample, and then you pop it into the PCR machine. You insert the temperature, and then you press go. And then you go have a coffee. 2 hours later, you come back, billions of copies. Understand? So that's one cycle, right? And you can see here, after one cycle, you would have 2 copies. And then if you denature, anneal, and elongate again, now you have 4 copies, and so on and so forth. Questions? Locate a gene of interest, check. Cloning the DNA, check. Next, we have to insert the DNA into a vector, right? But first, concept check. Remember to bring a calculator for Monday, okay? Don't ask me for one because I don't have it. Okay, let's do this. 4 cycles. All right. So you start with 10 copies. After one round, what number do I multiply by? 2. And then another round. 2. And then another round and another round. So that's 10 times 2 to the 4, which is 10 times, what's 2 to the 4? Okay. So basically, it is your starting copy, starting number multiplied by 2 to the n, where n is the number of cycles. Okay? No one's gonna do 4 cycles. At least, 15 to 20 cycles if you're gonna do PCR. Understand the math? Okay. Now, I was gonna tell you about the COVID test stuff, but that's gonna take up 10 minutes that we don't have. So we're gonna skip that. I'm not gonna ask you this on the test. All right. So no COVID testing. If you really want to know, ask me later. Next step. Yeah. So we located the gene, we cloned the gene. What's the next step, guys? That's right. You're gonna insert the gene into the bacteria. Okay. You're gonna encourage the bacteria to take the gene so that they can use it and make whatever you want them to make. In our case, we want them to make insulin. But before we talk about that, we have to understand a little bit of bacterial genome. Okay. Bacteria has circular DNA, which we already know when we learn about DNA replication, right? Our DNA are linear, their DNA are circular. In addition to their regular DNA, they also have additional circular DNA called plasmids. Okay, plasmids. Plasmids can be exchanged between bacteria, and plasmid can be taken up directly from a bacteria's surrounding. If, say, a bacteria die, and then another one comes along and see just random plasmid lying around, it will sometimes take it up and incorporate it into its own genome. Okay, so I told you this before. I think the plasmid kind of gives them special properties, right? Maybe the plasmid will give them resistance to an antibiotic. Maybe the plasmid will give them an extra protective layer outside of the cell wall, right? So maybe it does nothing, right? But the bacteria doesn't know that. It's gonna take up any plasmid in the surrounding and then basically see if it makes it better or not. Okay. So to understand how we feed the bacteria the gene that we want, we have to use something called restriction enzymes. Restriction enzymes. Restriction enzymes are actually a defense mechanism used by bacteria. Remember we talked about the bacterial phage, right? The virus would come and then it would insert genetic material into the bacteria. So bacteria actually doesn't like that. They don't like it when outside genetic material is being forced into them. And so they have these restriction enzymes that will cut them out. Okay? So if a virus try to insert its viral DNA into it, the bacteria will use the restriction enzymes to cut them out. Okay? How do they work? Well, these restriction enzymes usually have 4 to 8 base pair long recognition sites. They don't just randomly cut at different locations. It has to be a specific sequence that they recognize before they cut it. And these sequences are often complementary palindromic sequence. Let me explain. Yeah, well, it's online, right? So, yeah. So a palindrome in English is a word that spells the same forward and backwards. Okay? So madam is a palindrome. M-A-D-A-M. If you spell it backward, it's also madam, right? Okay, level, wow, kayak. And this is a very long one. Was it a car or a cat I saw? And if you read it backwards, it's also the same phrase. Okay? These are called palindromes. Same forward and backwards. Okay? A restriction enzyme will recognize complementary palindromes in the DNA. So over here, if you read from the top, GAATTC is the same as backwards on the other side. GAATTC. That's a complementary palindrome. Do you understand? All right? Maybe. Look, this is not a palindrome. If you have TTAC, hold on. See, that's not a palindrome in the context of what we're talking about. If you read it forward, it's TTAC CATT, right? If you read it backwards, it's also the same thing, right? But it's not a complementary palindrome. Because if you draw the complementary strand, right, this one is not the same as that one. Right? So when we are discussing restriction enzymes, the palindromic sequence is talking about the opposite strand. Do you understand? No? Okay, we'll do a few more examples. What's the missing half? Help me out, please. Read it this way. TTCGAA. Read it this way. Complementary palindromic. Understand? Yeah? One more. Is this palindromic or not? Yeah? Okay. If you read it this way, same as this one. It's palindromic, right? Okay, how about this one? Last one.
No.
No, right? Because if you write the other one is A, right? You understand? Okay. Why are we talking about this? These are sequences that will be recognized by restriction enzymes. Okay. And what do restriction enzymes do? They cut DNA. Not randomly, at specific locations. Okay. Here we have a restriction enzyme called eCore R1. It came from E. coli, okay? And then it will find GAATTC, and then it will cut it zigzag like that, and it separates. All right. Restriction enzyme is a big thing on the final exam. All right, so make sure if you don't understand, ask me now. Okay, we'll do some examples. Here are a bunch of restriction enzymes. You don't have to memorize the recognition site. I will always give it to you if I need you to know. What is important to see from this picture, though, is that sometimes the restriction enzyme will make zigzag cut. Zigzag cut, like that. And when you make a zigzag cut like that, you create sticky ends. It's actually what they're called. Meaning they can actually come back together because the stuff that are hanging out here, TTAA, is actually complementary to the AATT. So they would be able to come and find each other and then kind of like attach, like Velcros, like for the shoes for kids, like the Velcro. You can rip it off, you can put it back together, right? Easily. Make sense? Sometimes though, they would create a straight cut. Straight cut. Right, so like this, straight cut. And these are called blunt ends. Blunt ends. What we're trying to do is to insert the gene into a plasmid and then feed the plasmid to the bacteria. And for that purpose, we need to use the ones that create sticky ends. The blunt end ones are not useful for this application. I will show you how it's done in a bit. Just remember, these ones that creates the sticky ends, those are the ones that will allow us to insert the gene into a plasmid. Then we're going to feed the plasmid to the bacteria. All right, here is an example. We have a restriction enzyme called SMA1, small one, and the recognition site is GGGCCC. This is your DNA sample. How many times is it going to cut? If you have the notes, look at the ones on the paper. It's probably easier.
The bottom there. Oh, wait, okay.
How many times? 3 times? 2 times? Yeah. So all you have to do is look for GGGCCC. Okay. Remember, this is 5 to 3. So we look from the 5 to 3 strand. And GGGCCC is right here. Okay. So then look at how it's cut. It will cut right there. Make sense? Where else is it gonna cut? Okay, so you keep on looking for GGG, CCC,
Right there.
right? And then it's gonna cut again. That's it. If you do 2 cuts, how many fragments are you going to produce? How many pieces?
Eight.
Yeah, three. Okay, it's not rocket science, right? One piece, two pieces, three pieces.
Oh,
Okay, it's like having a piece of paper and then you cut twice, then you have three fragments. Do you understand? These are not going to separate because they are held together, right? By hydrogen bonds. Are we together? All right. See how many palindromic sequence you can find. Remember, it has to be at least four base pair long. Okay, four base pairs long. And I will come around and give you back your quiz. Not related. This question is just asking you where are the palindromes? At least four. Okay, let's see. This one is GC, right? That's a palindrome because if you write it backwards, it's also GC, right? That's one. Anything else? Okay, tell me to stop. Okay, if I hit one, tell me to stop.
eight.
Let's try again.
You can't do it.
It's okay. I'll just tell you where it is. Okay, right here. This one is GAATT. There it is. There it is. TTAA, right? And including the G here. See, there you go. You see that? Forward is same as backwards. Anything else? Here, tell me to stop, okay? Tell me to stop. Stop. Very good. There you go. Right? Make sense? I think those are it. I don't think there are more. Right? TTC, the middle one. Sorry, this way or this way? Left or right? This way? Left? Where? Here? PT? T TTCGA. No, because if you do it backwards, then it's like TCG. Yeah, like it has to be the same. Okay. I know.
Yeah.
Which one, huh?
BTAEG.
Oh, okay. Yeah, I see. Right. So they found another one. Very good. It's like here. TTCGAA. That's what you're trying to tell me, right? Okay, so that's TTCGAA. Right. So sometimes there are overlapping palindromes. Okay. You have another one? It's okay. We just keep this one for now. All right. But maybe you'll find more. Okay. The point is, will you be able to find them on the test? I'm not gonna ask you to find all of them on the test. Okay. I'll give you a sequence and then find two palindromic sequences that are 6 nucleotides long or something like that. Okay. Here's another one. Last one, and then we're gonna go for a break because we all need it. The DNA sequence below is treated with ECOR1 and HIND3. That's how you pronounce it. At the same time, how many fragments will be produced? Certainly sounds very happy about it. How many? How many? Five fragments? I actually have no idea. I haven't done this question for a whole semester. I don't remember the answer. We'll do it together. Okay. But do tell me what you think. If I put this on the test, I promise you I'll use big fonts, okay? So easier to see. Okay, so ECOR1, we are looking for GAA-TTC. GAA-TTC, right here. So that's going to be my first cut. All right. Please bring colors for the final test. You will need them. Is there another recognition site for ECOR1? Is there another GAA-TTC?
Yeah.
Yeah? right here, right? GAA, ETC. It's gonna cut it again. Make sense? That's all. You're just looking for the sequence, and if you find it, you're gonna cut it. Next, we're gonna find the recognition site, if any, for hint D3. And the recognition site is AAGCTT. Where is AAGCTT? Do we have any? Yeah, right here, right? Is that the only one? It is, right? I think it is. So how many fragments? Sorry?
Three.
4, right?
4.
Because you cut it 3 times, right? And take a piece of paper, you cut it 3 times, you have 4 pieces. All right, break time. 5 minutes.
Time to go.
Now we're gonna take a note on how exactly do we insert the gene into the plasmid. Now, forgive me, I have not write this note for quite some time for our whole semester. So let's hope I am gonna do an okay job here. Give me 1 second. Sorry. Inserting a gene into a plasmid. What are plasmid again? Can someone tell me what plasmids are? Yeah, very good. So plasmids are circular DNA that are extra in bacteria, and it gives them like certain powers, right? Certain properties. But we have the technology to create our own plasmid. Okay? You can actually buy plasmid online nowadays. Right? So when you buy a plasmid, the plasmid is going to be circular like that. And in the plasmid, when you buy it, it will tell you all the sequence, all the nucleotides in the plasmid. Now, to keep it simple, I am going to put a special sequence here in our hypothetical plasmid. I'm going to put T, 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 A. And then the other side would be like this, right? This is a palindromic sequence. And I am going to say that this is the recognition site for restriction enzyme 1. I just make this up, okay? Restriction enzyme 1. Which means when you take the plasmid and you mix it with restriction enzyme 1, it will cut the plasmid here. Okay? This sequence does not show up anywhere else in the plasmid. So it will only cut it once. Do you understand? Okay, one more time. This is a plasmid, and I put a recognition site in it for this hypothetical enzyme. If you mix the plasmid with the enzyme, it will look for this particular palindrome and it will cut it once. Okay. When you cut the circle once, when you cut the plasmid once, you open up the plasmid. Understand? Okay. So this is what we're gonna do. Okay. We're gonna have the plasmid is gonna be in a small tube like that. This is plasmid. I should write plasmid here. Okay, and you're gonna fill up just very small tube, just so that you have an idea of, the scale of these kind of reactions. And then you're gonna add restriction enzyme 1. It's just a random restriction enzyme that I make up. And afterwards, this is what it will look like. Sorry, give me a second. Sorry, almost there. Let me fix this up. I did it wrong. And make that a little bit smaller. Okay, there we go. Sorry. Now, the point is this. After you cut it, then now we have these sticky ends, right? TA here will bind to AT here. And the plasmid might close up. Yeah. You understand that part? I'm not going to ask you to draw any of these things on the test. But I'm hoping drawing it out now will help you understand it. So what we've done is basically cut the plasmid open. Are we ready for the next part? So this is 1, this is 2. What process do we use to clone the gene of interest? What process did we use to clone the gene of interest? Huh? PCR, yeah. Okay. So now, in this another tiny test tube, right? You would have millions of copies of the gene, right? Now, the gene is going to look like this. This is the gene of interest, insulin gene. And for this to work, and again, obviously, I'm oversimplifying things here. For this to work, the gene of interest would have to have the same recognition site flanking it. Flank means like on the sides. Okay, so can you listen for a second? In the plasmid, we have the TTTAAA, right? That's gonna be cut by the restriction enzyme. We need to have the same thing on the sides of the gene, but not inside the gene. Here is why. When I treat the gene with restriction enzymes, I want the restriction enzyme to cut the ends, but not the gene, right? If you also have TTTAA inside the gene, then you end up cutting up the gene and it's useless, right? If you cut the gene, you destroy the gene. You cannot make insulin anymore, right? So you need the same recognition sites flanking the gene. What does that do? Okay, just hold on a second. This one, if you add restriction enzyme, number one, the same one we have been using, now you will cut this and you will cut this. And it will look like this. Let me try to get this right. Um, there we go. And then it will cut over here. Did you say you ran out of space? There we go. Okay. So cut there. And in the end, what would you have? Well, you will have TAAA on one end. And then on the other side, you will have that. Now, I'll let you draw that in a second, okay? So this little bit just falls off. Doesn't matter. That little bit just fall off. But now we have created, what do we call this part here? That kind of hangs over. Sticky end, right? Okay. This sticky end is complementary to this sticky end, right? So this thing can actually goes here, and this is gonna go there because it's the same complementary sticky end. Okay, that's why we had to use the same restriction enzyme. If I use a different restriction enzyme and I get like, GGC or something, then the sticky end is not gonna stick together, right? If you cut things with the same restriction enzymes, you will create compatible sticky ends. So the whole point is you can mix this thing, and I'll let you copy that in a second. You can put this like right into there. Like it's not really showing properly, but like you see that? The T and the A will match up, right? Are you with me? And the same thing on the other side. It will match up, right? Okay, so copy what it is, and then I'll see who's trying to break in. So basically, if we mix this thing and this thing together, the green part will stick to the green part, the yellow part will stick to the yellow part, and then you would have your plasmid with the gene inside. Okay. What enzyme acts as a glue for DNA to stick together? Like this just goes in, but it's still not permanently fixed. What enzyme do we need to glue them together? Not that one. Good try though. Huh? I hear someone talking, but I don't know who's talking. Is it? Where? Which one is it? Oh, no, not the fragments. But yeah, what glue the fragments together? Which one? Not that one. It's okay. I'll just tell you. Right? Okay. So that will seal the gene, and then it will be okay. Now, again, I know it's a little bit overwhelming, and then there's a lot of information. But the key thing to understand here is if you cut the gene and the plasmid with the same thing, same restriction enzyme, then the ends will be compatible, and then you can glue them back together. Okay? Now, here is the thing, right? Listen carefully. When you add the glue, when you add the ligase, there is no guarantee that the gene has inserted itself in the plasmid. It is possible, and this is very important, it is possible when you add the ligase, you just end up gluing the plasmid back together without the gene. You understand that? Right? Because like when you add the glue, you might just glue this, or you might glue it with the gene inside. Right? There is no guarantee. It's a random process, right? So what you end up having is a mixture of plasmids. Plasmids with the gene and plasmids without the gene, right? So usually when you insert the gene, it's not just a gene that you insert. Right? You're gonna insert like a little bit of extra that cause the bacteria to make like a blue protein. Okay. So when the bacteria takes it out, when you start to grow them, then the bacteria will be blue, and you know that they have accepted the gene. And so that's one way to tell. But anyway, don't worry too much about that. Any questions so far about this inserting and then the gluing? Now, just very quickly, huh? If I have a plasmid here and it's TTA, let's do something else. If this is CCCGGG, and then you cut it, you don't have to draw this, you can just watch. If you cut it, then you might have CC, GG, C, D, and then GG. I always get this part wrong. G, G, GC, and then CC, like that. CC, sorry. Oh, that's why. So like this one pops open, right? But if I have the gene here and it's TTA, TTTAA, and that's not compatible, right? You understand? That's why you need to treat them with the same restriction enzyme. And you also need to make sure this restriction enzyme is not going to show up in the middle of the gene. Once again, if it does show up in the middle of the gene, you will end up cutting the gene as well. And then you will not be able to make the full protein. Okay? Next thing. This is a plasmid. Okay? It's a really basic plasmid. There are a couple things you need to understand from this plasmid. You know what? I'll do it on the PowerPoint slide so we can do it on the note here so I can explain better. Okay, that's just the name of the plasmid. When you order it, it's like, oh, I want to order PBR322, whatever, right? Now on this one, there is a ORI. ORI is basically the origin of replication. This is where transcription and translation are going to happen. Okay. And also where they will clone the plasmid. But basically it's gonna go and then it's gonna make all the genes in the plasmid. Okay. That's not so important. What's more important are these things. What are these things on the outside of the plasmid? Do you recognize any of them? Hmm? That's right. So on the outside, it tells you the restriction enzyme recognition site. If you take the plasmid and you treat it with E-core R1, it is going to cut it right here. If you treat it with PST1, it's going to cut it here. So on and so forth. Okay? So if you want to insert a gene, and your gene has these, TTTAAA, then you want to find the restriction enzyme that has the corresponding recognition site, right? Because sometimes, what if you don't have the TTTAAA? Then maybe I cannot insert using this restriction enzyme. I have to insert using something else. Right? So by having multiple restriction recognition sites in the plasmid, it gives you more option as to where to put the gene into the plasmid. These things on the inside, these are selection markers. Okay. So AMPR stands for ampicillin resistant gene. TETR stands for tetracycline resistant gene. Give me 1 second, okay? I'll come back, don't worry. So basically, if the bacteria takes up the plasmid, it will become resistant to tetracycline, which is an antibiotic. Normally, antibiotic kills the bacteria. But if it takes up this plasmid, it will be resistant to tetracycline, and then it will be resistant to ampicillin. Just copy it down first, and I'll explain further. Okay, so basically you have a bunch of plasmid now, a bunch of plasmid And then these plasmids, they are going to have the gene of interest. Okay, let me see here. They're going to have the gene of interest, which I'll just show in purple. And then you're going to feed it to the bacteria. This process is called transformation. You put them in the same tube, mix them together, and then there are things you can do to encourage them to take it up. But actually, they don't always take them up. So some will take it up, and then many will not. Bless you. So you mix the plasmid and the bacteria, and then you let them be for a little bit. And then after a while, some bacteria will have the plasmid. Some will not. You just don't want to take it up. That's fine. If we try to make insulin, which ones do we want to mass produce? Which bacteria do we want to grow? The transformed ones or the regular ones? Transformed ones, right? But here is the problem. They look the same in the tube. You can't tell which one is which. Okay? So when you try to grow them and on the dish, and then you see all the colonies forming, you cannot tell which one has the plasmid and which one does not. Do you understand so far? Okay, so using the color method, that's one way of doing it. But here I'm going to show you another way. Here's the main question. How do you tell which bacteria are transformed? Okay. This is where the selection markers are needed. Okay, so listen. Case one. To help you understand this, case one. Insert a gene using E-core R1. Then where does the gene go? Well, this is the cutting site for E-core R1 in the plasmid. The gene is going to go right here. Right here. Okay. Now, you're going to grow the bacteria. You're going to feed the bacteria with the plasmid, and then you're going to grow them in different plates. Okay, this, I know it looks the same. Let me try to do a little bit different here. Okay, so this is like a, it's called, it's an agar plate, okay? Agar plate. Okay, and this is just nutrients. And then I will have another agar plate. Agar plate is the brown dish that I showed you, okay? This is agar plate. Another agar plate. and it has nutrients plus ampicillin. Okay. So what we're going to do is, hold on a second. We're going to take all these things. We're going to take all these things. Oh boy. All right, we're going to take all these bacteria and then we're going to grow them here and we're going to grow them here. Okay. Remember, in the mixture, some bacteria has the plasmid and some don't, and you cannot tell. So when you put it onto the agar plate with just nutrients, do you think they will grow or not grow? They will grow, right? Because you give them nutrients, of course they're gonna grow. So everything will grow, okay? And you will see lots of colonies. Okay? It will grow everywhere. Okay. So you expect lots of growth. So this is actually our control. Okay. You want to make sure the transformation process didn't kill all of them. And you expect to see growth. What about the plate that has nutrients and ampicillin? Do you expect a lot of growth, no growth, or a little bit of growth? Only transformed bacteria can grow. And here is why. The plasmid has an ampicillin gene in it. So for the bacteria that has been transformed, not only do they have the gene of interest, they also has the ampicillin resistant gene, which is why when you try to grow them in the presence of ampicillin, the ampicillin, the antibiotic, will kill all the bacteria that do not have the plasmid. If they don't have the plasmid, they're not resistant to ampicillin, they will die. However, if they have taken up the plasmid, then not only do they have the gene of interest, but they will also be able to survive in the presence of ampicillin. Okay? So now everything that's growing are the ones that you want. Right? And then you can mass produce them. Right? And then they will make insulin, basically. All right. I will ask you a few questions here and see if you fully understand, and then we'll call it a day. A gene is inserted into the plasmid here, right? Using E core R1. So where does the gene go? Right here, right? And then you're gonna grow it on a plate containing tetracycline, which is an antibiotic. Do you expect them to have which I'm calling, like all of them will die, all of them will grow, or only small portion will grow? This is what we just talked about, right? Okay. Only small portion will grow. Okay. Why? Because not every bacteria will take up the plasmid. Only some will take it up. Okay. And if they take it up, they will be able to survive in the presence of tetracycline. Otherwise, they will die, right? Okay. It's not going to be, not everything will die because some will take up the plasmid. Not everything will grow because some will reject the plasmid, right? You can only survive in tetracycline if you have the plasmid. Next question. This time, the only difference is we are inserting using small one, okay? SMA1. And then you're growing it in tetracycline. What will happen? Well, if you insert using small one, the gene goes right here, right? By putting a gene right here, you actually interrupted the tetracycline resistant gene. Which means even if the bacteria accept the plasmid, they will not have resistance to tetracycline because the tetracycline resistant gene is disturbed by what you're inserting. So what will happen to the bacteria? They will all grow. Some will grow. They'll all die. What's going on here? They will absolutely all die. Okay? Look, without the plasmid, do they die or not die? Without the plasmid, do they die in the presence of antibiotic? Okay, antibiotic kills bacteria, right? You guys know that? Antibiotic kills bacteria. So without the plasmid, will they survive in tetracycline? No, right? They will all die. Without plasmid, they will all die. Okay. What about with the plasmid? Will they survive? Well, normally they would because the plasmid has the tetracycline gene for them, right? But now they won't survive because you've inserted something into the tetracycline resistant portion. So even if they accept the plasmid, they will still die. Okay? So nothing will grow. So if someone does this experiment, they did it wrong. Okay, everything died, nothing will grow. Okay, let's try a few other options here. If you insert with PST1, if you insert the gene with PST1, which antibiotic will you use to select for the bacteria? Should you be using ampicillin or should you be using tetracycline? Okay, anyone else? Here's the question. If you insert with PST1, do you select with ampicillin or tetracycline? All right, let's find out. Using PST1, where will the gene go? It's gonna go right here, right? If you pop a gene in the middle of the ampicillin-resistant gene, it will make it non-functional. Okay? What that means is, if you want to test whether the bacteria has accepted the plasmid or not, you will have to use the other antibiotic. You will have to use a tetracycline. Okay? If you use ampicillin to check whether they have accepted the plasmid, it will not work because everything will die. Nothing will grow. Even if they have accepted the plasmid, they will still die because you interrupted the ampicillin-resistant gene. Okay? But because the tetracycline gene is intact, you can use tetracycline to select. Okay, so use tetracycline to select. Okay, quickly, what about if you insert with BAMH? BAMH1. Okay, what are you going to use to select? Ampicillin, right? Because now if you put a gene in the middle of the tetracycline resistant gene, this will no longer work. Everything will die. Even if they have accepted the plasmid, they will still die. So you have to use ampicillin. Right? Okay, what about AVA1? If you insert the gene using AVA1, which antibiotic are you going to use for selection? Ampicillin, tetracycline, both, neither. Both is okay, right? Because if you insert into the alpha, using alpha 1 right here, you don't disturb that and you do not disturb that, right? And so if the bacteria takes up the plasma, they are resistant to both ampicillin and tetracycline. Right? And so you can use them as selection markers. Okay, let that sink in for a couple of days. We will resume on Monday. Remember to study very hard for your quiz on Monday. Be on